Good day, we are the Group 3 and we are here to present the characterization of bacterial culture. For the objectives, we have first, to enable the students to distinguish basic features of bacterial culture on agar plates, agar slants, and broth through the demonstration video. Second, to enable the students to recognize the advantages and limitations of cultural characteristics in the identification of bacterial species.
So for the first part is the characterization of bacterial culture in agar plates. Number 1 is pigmentation of colonies or color. Pyocyanin, a blue-green pigment, pyoverdin, a yellow-green pigment, and pyobin, a red-brown pigment, are the three most prevalent pigments in Pseudomonas aeruginosa colonies. As you can see in the picture, the blue-green one is the pyocyanin at a centromid agar. Next one is the pyorubin or the red-brown on a monkey agar. The third one is the pyoberdin. This one's the yellow-green on a centromid agar. So for the second one is the surface characteristic. So the Pseudomonas aeruginosa on an agar plate looks like a smooth, wrinkled, and contoured. It has a glistening appearance and smelled like grapes or tortillas. P. aeruginosa grows well at temperature ranging from 25 to 37 degrees Celsius, and its capacity to grow at 42 degrees Celsius distinguish it from many other Pseudom species. So the third one is the shape of colony. The shape of colony of the Pseudomonas aeruginosa is circular, large, opaque, and flat colonies with uneven edges and a strong fruity odor produced by the Pseudomonas. The sort of pigments produced by the organism will determine the color of the growth. Small round colonies are produced by water and soil isolate. So for the first picture, the red one is a blood agar and its shape is irregular. Next is the positive oxidase test. The shape is like a functiform. Next is the P. aeruginosa on TSA and its shape is circular. Then the next one is the Pseudomonas aeruginosa or S. aureus on STA and its shape is functiform. And lastly, the blue one is it's on a centromid agar and its shape is irregular.
The next characteristic is the size of colonies. Colonies can vary from large to tiny colonies less than 1 mm. The sizing of P. aeruginosa varies. Those grown in nutrient agar and blood agars are 2 to 4 mm in size, while the ones grown in McConkey agar are 2 to 3 mm in size. Margin of colonies. This characteristic describes the shape or formation of the edge of the colony when magnified. The P. aeruginosa grown in a McConkey agar possess an entire margin that is smooth and lack notches, while the one grown in a blood agar have a wavy with shallow indentions or an undulate margin. To present the remaining type of margins, other bacteria are used for characterization. Klebsiella pneumoniae, a bacteria which can cause various illnesses such as UTI and meningitis, possess a wavy with deep indentions on its margin known as lobate. Bacillus mycoides on agar plate is an example of filamentous margin. A Bacillus species growth on a Mueller-Hinton agar plate have an irregularly notched or serrate margin. A benign organism, Bacillus subtilis on a TSY agar plate, possess a curled or margin with parallel chains in wavy strands. Elevation of colonies. This characteristic describes the form and amount in which the colony rises on the surface of the agar. The Pseudomonas aeruginosa grown in a blood agar is flat, while the one grown in a soy agar plate have a low convex elevation. To present the remaining type of elevations, other bacteria are used for characterizations. The Escherichia coli or E. coli is a bacterium commonly found in the gut of humans and warm-blooded animals. An E. coli grown on a McConkey agar have an ambionate or a race with a thicker center elevation. A Mycobacterium fortitum, which belongs to a group of bacteria known as non-tuberculous Mycobacterium, grown on a blood agar, possess a raised or a thick with an abrupt edges elevation. Streptococcus pneumoniae, a bacteria commonly found in the nose and throat, which is grown on a blood agar, have an effuse elevation that is thin and unusually spreading. For the odor, we focus on the odor of the Pseudomonas aeruginosa. It is known to resemble a sweet, fruity, earthy smell. For the last characteristic of the agar plate, we have the color of surrounding medium. For this characteristic, we also focus on the Pseudomonas aeruginosa. Pseudomonas aeruginosa, grown on a nutrient agar, possess a green or pyoverding surrounding medium. The one grown on a McConkey agar and blood agar possess a dark red or pyorubin surrounding medium.
The characterization of bacterial culture in agar's land. First is the amount of growth, scanty or slight. The bacteria in the first agar's land is Escherichia coli. On the second picture, we can see the Micrococcus luteus, Rhodospirillum rubrum, and Bacillus subtilis. Second type of growth is moderate, which represents the Citrobacter frundi. Next, the abundant growth. In this agar's land, we can see the Trichoderma, which is type of fungus in agar's land. Next is none or no growth. This represents the Saccharopolyspora, which is an thermophilic bacteria. We can observe in this picture that in 20 degrees Celsius and 60 degrees Celsius, there are no growth of colony, while on the 30 degree to 55 degrees Celsius, there are abundant of growth of Saccharopolyspora. Next is the color of colony. Many bacteria create internal pigments that cause their colonies to resemble a specific color, such as yellow, pink, purple, or red. Many bacteria lack of pigmentation and appear white or gray. Next is the consistency. First is the butyrus or butter-like. This represents the yeast. This is a type of fungus in an agar's land. You can see the consistency of the colony that is butter-like. Second is the bacid, sticks to loop and hard to get off. This bacteria is Citrobacter frundi. Next is the membranous consistency. This is a sticky mucus-like that represents the Enterococcus bacteria in an agar's land. Next is the brittle or friable. The bacteria here in agar's land is actinomycetes that we can observe that the texture of the colony is dry and breaks apart. Next is the optical characteristics. First is the opaque that prevents the light from passing through them. This is a solid appearing. Second is the translucent. The bacteria here in the picture is E. coli, and some light passes through but not enough to permit visibility of objects viewed through growth. Next is opalescent, that like an opal in color. In this picture, we can see the fungus called Paracoccidioides and brasiliensis. Opalescent, that like an opal in color. In this picture, we can see the fungus that is called Paracoccidioides brasiliensis. Next is the iridescent, that change in color in reflected light. Next is fluorescent. When we say fluorescent, it is the emission of light by a substance that has absorbed light. In this first picture, we have the bacteria Pseudomonas aeruginosa and Pseudomonas fluorescens in a ceterimide gas. Next picture is the E. coli. Next, dull. In this picture, we have the E. coli. Then, last is the glistening, which shines with a sparkling light. In this agar's land, we can see the Bacillus cereus. Form. Number 1 is the filiform. We can see the bacteria of E. coli in the agar's land. We can compare the filiform from spreading. In this picture, we can see the spreading is occupied at the bottom of test tube, while on the filiform, we can see only growth of bacteria on the straight line position. While on the spreading, we can see that on the bottom, there is occupied growth of colony, and this represents the bacteria Bacillus subtilis. The spreading is called also as effuse. Next, echinolate. In this picture, we can see the Micrococcus luteus. The echinolate is like a spinning pattern of growth. And next, the beaded. We have the organism type of fungus which is called Geotrum trichum candidum. You can see the colony growth of fungus are shaped like beads, which called beaded. Next is rhizoid or root-like, and the arborescent. We can compare the difference of these two forms in their pattern. We can see that the rhizoid type is like a roots, while on the arborescent they are branch-like tree. So, unluckily, we didn't found an actual picture of rhizoid form. And in the arborescent, we represent the bacteria of Lactobacillus acidophilus or Lactobacillus acidophilus. So, all in all, these are the form of pattern of growth in agar's land, which is the filiform, echinolate, beaded, effuse, arborescent, and rhizoid.
The bacteria that is presented on the slide is Serratia marcescens. The odor is difficult to describe. It is often related to a fishy or urinary smell. Different observers may have different perceptions of the odor. Medium change. As for the color change, the species name marcescens means fading away, suggesting that the fading of the red color is characteristic of the species. A difference of 2 degrees Celsius have resulted to varying pigments. The color lessens as the temperature increases.
Characterization of bacterial culture in nutrient broth. Surface growth. There are 4 types of surface growth in nutrient broth: the ring, pellicle, flocculent, and membranous. First off with ring. A ring-like structure is visible adherent to the glass on the surface of the liquid medium. Next is pellicle. Mycobacterium smegmatis growing on the surface of the media forming a pellicle. A pellicle is a growth in the form of a thick film on the surface of the broth. Next up is flocculent. Bacillus subtilis produces flocculent growth in liquid. Flocculent is characterized by a flaky clump growth of various shape floating in the liquid medium. Lastly, for the surface growth in a nutrient broth is the membranous. Pseudomonas aeruginosa forms a membrane on the surface of a broth culture. Membranous is a thin fluid sticking together on the surface resembling a membrane. The second characterization is the subsurface growth. Turbidity, when the medium appears to be cloudy. This happens when the organisms diffuse uniformly throughout the broth. Granular, small particles are visible floating in the medium. Next is flocculent. Flocculent is when the small masses of various shapes could be seen floating in the broth. And lastly is flaky. Flaky is the large clump of particles that could be seen in the liquid medium. The third characterization of bacterial culture in nutrient broth is the amount of growth. None. No organisms present, no visible change in the texture of the medium. This could also be noted by the clear appearance of the liquid media. Scanty indicates that the organisms was only present in the initial sample inoculum. Shows slight turbidity or cloudiness, meaning there is some bacterial growth in the medium. Moderate indicates that there is a significant amount of organisms present. And lastly is abundant, indicates that the organisms was present in all areas. The fourth characterization is the sediment, starting off with granular. Small granules settle at the bottom of the medium. Next is flocculent. Small masses of growth of various sizes could be seen. Next is flaky. Large particles resembling flakes that settles at the bottom. And lastly is viscid. Viscid is the rising sediment that forms a swirl. And the final characterization of a bacterial culture in a nutrient broth is pigmentation. This could be identified as either none or any presence of pigmentation or changes in color in the liquid medium.
Characterization of bacterial culture in agar slants. Amount of growth. The first one is none. This can result if the source culture is dead or if the correct medium or growth conditions for the strain weren't given. Next is scanty. It is a minimal or small amount of growth. The slide shows an agar slant culture of the fungus Sporothrix kenki at 37 degrees Celsius. The second picture is a Saburao dextrose agar slant culture of the fungus Paracoccidioides brasiliensis grown at 37 degrees Celsius. The next is moderate. The slide shows a picture of the Trichophyton agars, and the next one is the Saburao dextrose agar slant culture of the fungus Cryptococcus neoformans grown at 37 degrees Celsius. The last is the abundant growth. The slide shows a Saburao's dextrose agar slant. Form of growth. Unfortunately, there weren't a lot of pictures available online, and these were the only ones that I found. The first one is filiform. Next is beaded, with its example, Geotrichum candidum. Next is papillate, villous, and last is arborescent. Next characteristic is liquefaction. Liquefaction is the reduction of gelatin culture medium to the liquid state that is caused by enzymes produced by bacteria. The first one is crateriform, lapiform, infundibulum, saccate, and the last is stratiform.
That will be all. Thank you so much for listening.